My name is Elisabeth, and I'm a PhD student in neuroscience here at RCSI. I am from Malmö, Sweden, and I came to Dublin about 3 years ago to start my PhD. Before this, I studied at Lund University in Sweden. There, I completed a degree in biotechnology engineering, and as part of my degree, I also spent a year abroad in San Francisco, where I studied neuroscience. During this time, I became very interested in research, and especially research on neurological diseases. And that's why I decided to do a PhD in neuroscience. My research focuses on the disease amyotrophic lateral sclerosis, also known as ALS or motor neuron disease. It is a rare disease, but became known worldwide very much because of the renowned physicist Stephen Hawking, who lived with ALS. You may also have heard of the ALS Ice Bucket Challenge that became viral back in 2014 and has further helped raise awareness of the disease. ALS is a disease of the nervous system. It affects a specific type of nerve cells called motor neurons. These are the neurons that connect our brain with our muscles and control voluntary muscle movement. ALS causes the motor neurons to degenerate and die so that the muscles no longer receive signals to move. This in turn causes the muscles to weaken and waste away. ALS usually begins in adulthood and advances rapidly. As the disease worsens, people with ALS lose their ability to move, speak, and ultimately breathe. There is currently no cure to stop or reverse ALS, and most people with the disease only survive for a couple of years. For some, ALS is inherited, but in most cases, the cause is not known. ALS research is important to increase our understanding of ALS and what causes it, so that new and better treatments can be developed. The goal of my research is to investigate the cellular and molecular mechanisms that are involved in the disease so that better treatments can be developed in the future. To better understand how we study ALS in the lab, let's first have a closer look at what happens to the motor neurons in ALS. One of the major hallmarks of ALS is the clumping or aggregation of proteins inside affected motor neurons. These ALS-specific protein aggregates, they appear toxic to the cells and can cause injury or cell death. To mimic the cellular processes in ALS, we culture neurons in the laboratory and then expose them to synthetic versions of the toxic ALS proteins. The proteins are taken up inside the neurons where they aggregate, causing the cultured neurons to degenerate and die. We can then use this cellular model of ALS in our experiments to investigate molecular mechanisms underlying the disease.
My name is Stephen Fagan, and to give you some of my background, I did my undergraduate degree in biotechnology at DCU. I moved to Bristol for a master's degree in molecular neuroscience, and then back to Dublin to do my PhD in Trinity College. Since then, I've worked as a postdoctoral researcher at the University of Cambridge and now Royal College of Surgeons. Here in RCSI, I research epilepsy. Epilepsy is actually a group of brain disorders characterized by repeated seizure activity. In our brains, there are about 100 billion interconnected neuronal cells, all communicating through electrical signals, like the motherboard of a computer. A seizure is when there's a surge of electrical activity all happening at once, and this can short out the brain. A seizure can be detected with sensors placed on your head, and you can see here under normal conditions, the electrical activity is calm and pretty repetitive. During a partial seizure, you can see the strength of the electrical activity increases significantly in one part of the brain. And during a general seizure, the electrical activity is increased all over the brain. About 50 million people worldwide have epilepsy, including 37,000 people in Ireland. Symptoms of a seizure include loss of consciousness, anxiety, staring blankly, muscle weakness, and sudden jerking of the limbs. Luckily, there are many great medications for the treatment of epilepsy, and these work in the majority of cases. However, about 30% of epilepsies are currently drug resistant. So we research epilepsy to better understand what causes it and how we can best treat people who have it. To do this, we use a cell culture system. Cell culture allows us to study a single type of cell, in this case neurons, under controlled conditions. That allows us to induce epilepsy analyze how the cells react, and to test drugs. Here is a short video showing how we culture neurons. Cell culture is carried out under sterile conditions in a laminar flow hood. Air is blown from inside these hoods towards you to stop any bacteria from floating in. The surface is cleaned with 70% alcohol before use. Everything is sprayed with alcohol before being put in the hood to make sure that they are sterile. Cells are cultured on plastic dishes to give them a hard surface to grow on. Neuronal cells in liquid media are carefully seeded onto the cell culture dish. Neurons are very small, and each well in the culture plate contains over 1 million neurons. These neurons need to be kept under optimum conditions and will therefore be stored in an incubator that maintains a constant temperature of 37 degrees Celsius and an atmosphere of 5% carbon dioxide. This is just like the environment found in your body and brains.
To study these tiny living cells and check how they are growing, we use a light microscope, which allows us to see a magnified image of the cells. A couple of hours after seeding the cells, we can see hundreds of neurons under the microscope. But before we look closer, let's remind ourselves of what a typical neuron looks like. A neuron can be divided into 3 distinct parts: its cell body, dendrites, and axons. The cell body also contains a nucleus. Extending from the cell body are the dendrites, which look a little like tree branches. These are connected to other cells and function to receive information. The information is then carried to the cell body and along the axon to the axon terminals, where the information gets transmitted to the connected cells. Now let's zoom in on the microscope picture. During the first day in culture, the neurons are very young and what we refer to as immature. At this point, we can only distinguish the round cell bodies, but over the next couple of days, the immature neurons will grow and develop axons and dendrites and start to connect with one another. After 4 days in culture, we need to change the liquid media on the neurons. Media contains all the nutrients and growth factors for the cells to stay healthy and glucose as an energy source. Healthy cells will quickly eat up this media, so it is important to apply fresh media regularly. Looking at the cells in the light microscope, we can now see how the neurons have started to grow dendrites, axons, and also started making contact with one another. We then put the cells back into the incubator for another few days. It is always important to remember to clean up after using the hood to make sure it stays sterile for the next time. Here, the neurons have been cultured and allowed to mature for 8 days. At this stage, they have developed their dendrites and axons, and each neuron is now connected to many others. What we see is a dense network of communicating neurons, which are now ready to be experimented on. To generate a cellular model of ALS, I add the toxic ALS proteins directly to the culture media, and the neurons then quickly take up the proteins. I then use advanced microscopy techniques to investigate the effect the proteins have on the cells. One of these techniques is laser confocal scanning microscopy. Confocal microscopes use laser light to produce high-resolution images of cells in both 2 and 3 dimensions. This allows us to study cells in detail and learn what goes wrong when they become diseased. So, let's have a closer look at the cells under the confocal microscope. Here you see a 3-colored image of cultured neurons exposed to ALS proteins. We can actually split this image into 3 separate ones. In the image to the left, we see the cell nucleus in blue. So these little blue dots are all nuclei. When a neuron dies, its nucleus shrinks, so all the small and very bright blue dots that you see, they belong to dead or dying neurons. In red, we see the cell bodies and also the dendrites extending from the cell bodies. And the green clumps that you see in the image to the right, they are the toxic ALS proteins. If we merge these 3 images into one again, you may now be able to identify single neurons, each containing a cell body, a nucleus, and connecting to many other neurons. And if we have a look at the toxic ALS protein aggregates that are shown in green, we can now see how these locate to the shrunken bright blue nuclei. This shows us that the neurons which have taken up the proteins have died, meaning that the cellular model of ALS is working. I then use this model to investigate the molecular mechanisms that occur in the cultured neurons, to learn what goes wrong in neurons affected by ALS and how to protect and rescue them from the disease.
A few years ago, researchers here at RCSI discovered that the levels of a newly identified molecule increase in the blood leading up to a seizure. These molecules, or tRNA fragments, were found to be released by neurons and can act as a warning sign that a seizure will occur. We call this a biomarker. tRNA fragments are small bits of genetic material copied from DNA and have been shown to affect how cells react to stress, inflammation, and the regulation of gene expression. My research is trying to identify what these tRNA fragments do to epileptic neurons. I add these molecules to our neuronal culture, induce a seizure, and see how they react. After adding my tRNA fragments to the neurons, I first confirmed that they got into the cells okay. Using a confocal microscope, I can see the neurons here, in green, and the tRNA fragments here in purple. It looks like they overlap. I then go to a different microscope to look at the electrical activity of the neurons. For this, we use a dye that binds to calcium, and when there's electrical activity, the dye shines much brighter. Here are the neuronal cells, calm, not much activity. And now we induce the seizure, and you can see how the cells are much more active.